Have you ever wondered the medicine which we are consuming to cure a disease? How does that drug comes into existence? Who did it? That I am going to discuss today in this drug discovery chapter.
Thing is, we have to find out different compounds that can be processed into different steps that it will go and approved by FDA and will be available as a drug in the market. So before the market, when the drug will go, different steps are there that is unknown to others. That unknown steps we have to study in pharmacy. That's why we came here to study pharmacy. So first thing, that part is comes under pharmacology, or that we know, that is called as R&D or research and development. That research and development comes under pharmacology. So what it will do, it will tell us how we have to choose a compound against a disease, and that compound is going through different steps and it is introduced as a drug into a market as a drug to use by the normal consumers or people who are fighting against a specific disease. So how the drug comes to market, you don't know that thing. So compound to drug, it goes through a few steps. That two steps or that steps are classified into two main parts. One is drug discovery. Second part is drug development. From the name only you can understand, discovery means we have to find out a new compound or new drug that is not available in the market. So that can be anything or any source. That can be, that compound can be synthetic, that means prepared in lab, or it can be natural, that is already present in different natural things, especially the plants. And the discovery of drugs, it can be sometimes by intentionally, it is comes by a long research and time of hard work by a particular researcher to introduce a new drug into the market. And sometimes the drug discovery may be accidental, in that generally one pupil or one researcher is generally started their experiment to find out a particular compound, but by accidental they come across a new compound that can be act as a drug against a specific disease. So in drug discovery and drug development, different steps are there that we are going to discuss one by one into the next slides. First, you have to remember, for that drug totally from that compound to the marketed drug, it will take 10 to 15 years minimum, and it go through different steps, and that steps are classified into two parts. One is drug discovery, second one is drug development. That development we will discuss later. First, go through the discovery. In that discovery also, four steps are there. First one is target identification. Second one is target the second one is target validation. Third one is lead identification, and the last step is lead optimization. So target identification, target validation, lead identification, and lead optimization are the part of drug discovery. And in drug development also different phases or different steps are there that I will go to discuss when I will come across or I will discuss with development. Let's focus first into the drug discovery. The first step of drug discovery is target identification. So by the name only you can understand we have to identify the target for which we are inventing or we are discovering the drug. Thing we have to in that world, one is specific disease is increasing day by day, but there is nothing or there is no available drugs in the market that can be used to effectively treat that disease. So now to treat that disease, researcher or the pharmacist, they started to discover different compound. From that different compound they try to discover or try to produce a specific drug that can be effectively used against that disease to overcome that disease. So first we have to identify the disease against which we have to introduce a drug into the market. So first in the target identification, we have to target or identify a specific disease against which we have to invite or invent a drug. That step is called as target identification. That how we can know? First we have to know how that disease is causing that causes of disease we have to check out, so that we can effectively find out the drug. The second one is the involved tissue, organ or receptor that is generally affected by that disease. That also we have to identify into the target identification part. The second point or second step is target validation. So in target validation, by the name only you can know. Validate means we have to identify whether the collected information are right or wrong. So in that procedure, what generally takes place is in target identification, what we are getting or collecting information about disease like disease cause, what the tissue receptor and the tissue receptor or organ affected by the disease, what can be the possible causes and possible mechanism of action by which that disease can be overcome or disease can be treated. All that information that we have collected in target identification, in target validation, we have to validate whether all that information collected during the first step is true or false. After validating the true statements, from the true statement we have to take some key points that we have to note out into the next point, so that it can be keep into our mind to introduce a new drug that can be effectively used against that disease. That step is called as target validation. Third one is lead identification. So after informing different information that I have already discussed into the first step, we have to validate whether it is true or false. After validating all the 2 points, we have to make some key points which is we have to keep in mind to introduce a drug into the market. So remembering all that true data that is collected during target validation and target optimization or target identification, all that data is recorded into that lead identification. In lead identification, what we have to do, we have to find out some compounds that have same opposite property of that disease, so that all that compounds that have opposite properties of that disease, we can use that compound in treating that disease or to treat the symptoms of that disease. So to find out the targeted compounds is the main purpose of lead identification. In that method, generally 5,000 to 10,000 compounds are shortlisted, which are going to be the tested compound that will go through the next step for lead optimization. In lead optimization, what it will happen after identify the targeted disease, their cause, and their mechanism of action, and some compounds which can be used, which are having opposite property of the disease, can be used to treat that disease. That compounds have to be optimized, and that optimization is the main purpose of lead optimization step. That compounds 5,000 to 10,000 compounds that can be optimized by different methods, can be optimized by different methods, like what can be the possible mechanism of action of that compound. Second one is what can be the possible structure, action, activity relationship of that compound. What is the pharmacokinetic, physical, chemical properties of that drugs or that specific compound. All that we have to study, or we have to check or optimized in that leading optimized chapter or leading optimized step. We have to check out, and that is the main purpose of lead optimization. So by that I have finished off the drug discovery part. After the drug discovery or finding out a specific compound, in lead optimization, that compound is optimized and is now ready to be tested as a drug into different testing bodies. It can be first animal, then after that pupil. That all testing condition or testing steps, it comes under drug development phase. So we have to now study what are the different steps involved in drug development. So the steps of drug development. First step is pre-clinical trials. Second one is investigational new drug application. Third one is clinical trials. Fourth one is new drug application, which are generally done by Food Drug Administration body. And the fifth and last part is pharmacovigilance or post-marketing surveillance. That all steps comes under drug development. That all steps I am going to discuss into the next steps or next slides. The first step comes is preclinical trials. So after inventing or after discovering some compounds, we have to now check whether that drugs or compounds can be used as drug or not, or whether that compounds really they have therapeutic effect or not. What is their efficacy rate? What is their potency rate? What is their toxic effects? What are their pharmacological effects? All these things we have to test into some volunteer or some experimental bodies to get to know whether that drugs are effective or not. So for that we can take pupil or animal. But before going to the testing in pupil, that drug compound should be tested in animal so that the drug cannot cause serious, effective, harmful damages to pupil. So before testing into the pupil, we have to search whether that compound are harmful to people or not, so that people should not harmed by that compound. So first invented compound we have to test it into the lab animals to check its efficacy, potency, toxicity, pharmacological action, etc. So in that preclinical trials, different animals are used. That animals can be rat, rabbit, guinea pig. Sometimes dogs and cats are also used. But the most of the time, rats, rabbit, and guinea pigs are used because they have psychological, physiological, and anatomically similarness with human. So they make more resilience, or they make more comparability with, or more matching with human, so that we can check whether the drug can be affected into the similar type of psychology or physiology or anatomy in that animals. So these trials can be into two parts or in two types. One is in vitro, second one is in vivo. In vitro means outside the animal. That means for the testing or trials of the drug, here the whole animals are not taken, only the some tissue or targeted organs or targeted receptors of that animals are collected, and that drug compound are tested on all that tissues that is collected from the animals. And in vivo, that drugs compounds are tested inside the animal by injecting or administer the drug into that animal to check the changes inside the body of animal, so that we can understand the drug is acting or not. That whole process generally takes 5 to 6 years of times. So the next step in drug development after preclinical testing is INDA or investigational new drug application. So when that drug or that compound is tested into the animal, into the preclinical testing, some result comes out that generally differs about the drug pharmacology, pharmacokinetics, toxicity, efficacy, potency, therapeutic index, etc. So all that result we have to send to INDA committee, who will discuss or check out all that result, and after seeing the validation of that result, and after discussing about themselves, and after deciding whether all that collected informations are right or wrong or genuine, they will give their verdicts. And if all that data is giving the true value or the true result of the testing, then that INDA committee will give the approval of the drug to test that drug into humans. So that approval or application is called INDA or investigational new drug application. After the approval of INDA, that drug can be used or that drug can be tested into the human. That comes under the next steps, which is clinical testing or clinical trials. The next steps after IND approval is clinical testing. So when the drug got investigational new drug application approval, that drug can be tested into the human volunteers. And that testing in human volunteers of a particular drug is comes under clinical trials. Now, according to the numbers of human volunteers and the specific part or specific reason for the drug testing, depend upon that clinical trials have four phases. One is phase 1, phase 2, phase 3, and phase 4. And phase 0 is also there. That 0 is called as preclinical trials that I have already discussed before IND or investigation and new drug application. So we have to now go through different phases of clinical trials. I already discussed about phase 0 or free clinical testing. Now we have to discuss about phase 1, phase 2, phase 3 and phase 4 of clinical trials. So for that you can understand you can understand the clinical trials by a simple box or by a simple list. That is the phase, that is the number of human volunteers, and that is the reason or the parameters that we have to try or we have to test into the human body, and what is the time duration of all these tests. So first I have already discussed about phase 0, that is preclinical testing. It generally takes 5 to 10 years, then INDA, then it comes to clinical trials. So first one is phase 1. In phase 1, generally 30 to 100 human volunteers are taken to test the efficacy, potency, pharmacology and toxicology of that drug into that volunteer, and the number is 30 to 100, and it generally takes 1 week to some months. And most of the drug, almost 70% of compound, pass that phase 1 clinical trials step. Then the next step is phase 2. Here 70% drugs go through the second step. That is second step or second phase. Generally the drug is tried or tested into some human volunteers. The number of the human volunteers are 100 to 500 volunteers. Here what we have to check, we have to check the dose, safety of human, how many people are getting cured by the use of the drug and how many are affected by the side effect, that we have to check into the phase 2, and for that generally 100 to 500 human volunteers are taken. For that steps it generally takes some months to 2 years. In that step, out of that 70% drug, 30% drug got past that second step or phase 2 clinical trials. Then that 30% of compound is go through phase 3 of clinical trials. In phase 3 clinical trials, generally 1000 to 5000 human volunteers are taken as the testing human volunteers. In that what we have to check, we have to check here drug interaction, safety, therapeutic index, toxicology, etc. And that steps generally takes time of 4 years to 5 years. So and in that 10% drug got selected. That 10% now will send to the national or new drug application commission to get the approval. Next step is new drug application. So when the 10% drug got passed through the clinical trials phase 3, that 10% drug are sent to Food Drug Administration Committee for NDA application. Now that FDA will check all that result comes from the clinical trials phase 1 to 3, they will validate the result, and after the validation of result that all that collected information are true and validated, they will give a approval of new drug application. By that application approval, that drug can be sent to the market and can sell into the market, so that normal consumer can buy that drug and can use to treat that particular disease. That step is called as new drug application, and that is generally approved by Food Drug Administration Committee. After that, drug go through the market and normal consumer can buy that drug and can use to treat that particular disease. The last steps of drug discovery is drug development is generally the same step of clinical trials phase 4. So clinical trials phase 4 and the 5th steps of drug development is same. In that, what it will happen after the approval from NDA, the drug will go to market, and from market different consumer or patient will collect that drug to treat the disease. Now, how many people are taking the drugs? How many of them are getting cured? How many have side effects due to the drugs? How many got survived from that disease? And how many got died from that disease? All that information are taken into that phase 4 of drug development. That drug development phase is called as pharmacovigilance. So that was the last stop or last step of the drug validation, to check whether the drug is acting or not into the normal consumer or patient. Then it is going to check how many percentage of people got cured by the drug and how many got affected or died by the drug. Then it will get decide whether the drug will be in market or not. So all this comes under the research or production of a drug, that will go through two main parts, that is drug discovery and drug development. In drug discovery, I told you 4 steps was there: targeted identification, targeted validation, lead identification, lead optimization. Then it comes to the drug development. In drug development, first step was preclinical trials. Then after preclinical trials, it will go to IND approval. After IND approval, it will get tested into the human body. That is clinical trials, and that clinical trials comes 4 steps: phase 1, phase 2, phase 3. After completing of phase 3, the drug will go through the FDA or Food Drug Administration for the approval of new drug application. After the new drug application approval, that drug will get marketed and that consumer can buy that drug. After that marketing of drug, pharmacovigilance or phase 4 clinical trials will comes under. Here a team will come and will collect all the data from different medical stores and market, that how many drugs are bought in a specific time of period, among all that patient how many got cured, how many got died, and according to that they decide whether that drug is acting or not, or whether that drug will be in market or not. So this is the whole drug discovery chapter, that you don't generally know how that drug is produced or drugs comes into the picture of a mind. So that all are the steps I have discussed easily by a simple diagram or a simple chart that you can easily understand. Go through all these things, and the links of all these notes are given below into the description of video. Thank you, and go through this.